Okay, so we're going to talk about the retina. Okay, now the retina is the light-sensitive pigment at the back of your eye, back of the mammalian eyes. We'll talk about the human retina in particular here. And it's quite strange. The light-sensitive, the light-detecting cells are actually down here at the bottom. All right, so the light that comes in, you can see the light actually comes in from this direction, and so it has to pass through some of these nerve cells before it actually gets to the light detecting cells. That's how it's arranged. Right, so at the bottom of the retina, we've got the pigmented cells here, and that is just sort of light bouncing around the back of the retina. So that is the pigment melanin. It looks black, and that's fairly important in people who can't produce melanin. In albinism, they have problems with their eyesight for that reason. Okay, now you can see there are two kinds of light detecting cells. There are the rod cells and the cone cells. Now the rod cells are they absorb, they detect light at low intensity and over quite a wide range of wavelengths, colors in other words. Okay, they absorb best apparently in the blue-green part of the spectrum, but they do the red also, and they've got one light-sensitive pigment there. It's called rhodopsin. It actually has a vitamin A molecule in the rhodopsin molecule. It's a protein, but it's the non-protein part of it. The vitamin A molecule is the bit that's responsible for absorbing the light. Now, the cones, and I've drawn them in red, obviously they're all the same color really, the cone cells, and they do have a slightly different shape. That's why they're called that. They are responsible for detecting color. They work at a much, they need higher light intensity. In order to work. So that's why you can't distinguish colors very well at dusk or in light, and when there's low lighting, you can't distinguish colors, and that's because the cone cells need higher light intensity in order to work. Now the cone cells, there are 3 different receptors, okay, and the 3 different receptors are called opsins. They also contain vitamin A as a light detecting part of the molecule, but you have opsin, there's one which is opsin S, and you have that detects short wavelengths, blue. Then you have medium, that detects longer wavelengths, which are green, the green part of the spectrum, and then you have the one opsin L, which is longer, that detects red light. Okay, so you can detect those 3 different colors, and the way in which they are stimulated enables you to see the whole spectrum. So you probably remember from physics, if you get green and red together, you shine green and red light together, you end up with yellow. That's what it looks like to our eye. So when you see yellow light, it's because you're getting some stimulation of the medium one, which is detecting green, and some stimulation of the red one, and you perceive that as being yellow. So that's how color vision is managed with the cone cells. Now we'll just talk about the connections. Okay, so now we said that rod cells are better at working at low intensity, and if you look at these, so these are the bipolar cells, and they connect the light sensitive cells here, the rods and the cones, through the ganglion cells to the optic nerve. Well, you can see here, I've drawn sort of stylized, but I've got 3 different rod cells, and they're all impinging on the same bipolar cell. So that means, you know, you don't, if you're getting a threefold multiplication of the signal in effect, really, there. So rod cells are good at detecting low light intensity. Wherever you look at the cone cell connections there, so if you look at that one there, it's usually the case that each one cone cell abuts onto one bipolar cell. So unless you've got a very strong stimulation of that cone cell, it's not going to be enough to trigger a nerve impulse in the bipolar cell. You've only got one of them to do it. So that partly explains the difference in the sensitivities and the light intensities of the rod cells and the cone cells. Right. Now we need to also talk now about visual acuity here. So I'll just get rid of this. Okay, so visual acuity is basically how good is your eye at resolving 2 points. Okay, so then you've got 2 points of light there. If they're very close together, your eye will just perceive it as 1 point of light. Now, if they're a long way away, it will be able to distinguish that it is actually 2. So it's like resolution, really, is another way of thinking about it. It's how good at resolving the image it is. Now, rod cells, they give poor acuity. And the reason for that is, again, it's due to this sensitivity things. You've got 3 rod cells which are abutting onto one bipolar cell. So your retina can't tell whether it's that one, or that cell which is being stimulated. So it's not very good at telling exactly where the light is coming from. You get much better visual acuity with the cone cells. Good acuity, because each cone cell is only attached to one bipolar cell. So if this bipolar cell here, that one there, is being stimulated, then you know it's because it could only be that cone cell, not any others. So that you can tell exactly where the light is coming from. Now, where are the rod cells and the cone cells located? Well, the cone cells they're packed in principally into the central part of when you're looking straight ahead, and it's called the region on the retina is called the fovea. It's in the central part of the retina. So that you get the best visual acuity in the central part of the retina. So when you're reading a book, you're you're focusing light on the fovea and you're getting good resolution there. You get less good visual acuity in the periphery of your vision. However, the fovea is there's only cone cells in there. There's no rod cells. So that means it's not very good at the fovea is not very good at detecting low light. And you probably experienced this if you look in the sky at a dim star. If you look straight at it, at this dim star, you can't see it very well. If you just look to the side of it, then very often then you can actually detect that it's there. And that's because the light is then falling away from the fovea onto an area of the retina where there are a greater number of rod cells. Just a word about these the pigments. Let's talk about this. So in the rod cells, the actual light detecting pigments, so the receptors, which I've said also proteins with vitamin A in each case. Well, in the rod cells, you have rhodopsin, and that is located on chromosome 3. The cone, you have 3 opsin genes. Sorry, opsin genes, and they're all located on the X chromosome. And you're probably aware that most color blindness is X-linked, and the reason for that is because the 3 color receptors are on the X chromosome, and so if you get a deletion of one of those 3 things, it means you can't you're not very good at detecting either the short wavelength, the medium wavelength, or the long wavelength. And you're probably aware that the most common kind of color blindness is when you don't have either the medium or the long, and the medium detects green light and the long detects red, and very often those people have difficulty distinguishing between green colors and red colors. These three opsin genes, if you look at them, they are all very similar sequences, and it's fairly obvious that they've arisen by duplication of the original gene, and then there's been some kind of mutation that absorbs a different wavelength of light. The rhodopsin genes, they're also, there is some sequence homology to the opsin genes there as well. They obviously come from a common ancestor, but it's a lot further back. The rhodopsin and the opsins, they diverged from each other a long time ago, whereas these three opsin genes, these are all arisen quite recently in evolutionary history. Okay, so just talk about, there's obviously a lot of synapses there, you can see. So you've got the bipolar cells are forming synapses with the rod cells and the cone cells, and they're also forming the synapses with the ganglion cells, and the ganglion cells then obviously go into the optic nerve. Those synapses, there are different neurotransmitters. Acetylcholine is involved, but some other neurotransmitters are also involved as well there. Okay, so there's the retina. You need to know it's an example of a sensory organ, and we have the special sensory cells, the rod cells and the cone cells, and the associated nerve cells with them, which are required for sending the messages onto the brain.